as you have seen, all the DNA polymerases, they require a primer which has a free 3' hydroxyl group. So these DNA polymerases, they cannot initiate synthesis of new DNA strand de novo. They by themselves, they cannot start synthesis. Then how they synthesize new strands? So to accomplish this, the cell takes advantage of the ability of RNA polymerases to do what DNA polymerases cannot. That is, to start new RNA chains by themselves. That is de novo synthesis of RNA chains. Primase is a specialized RNA polymerase that is dedicated to make short RNA primers, about 5 to 10 nucleotide length, on a single-stranded template DNA. So when a single-stranded template DNA is formed, these two single-stranded templates are formed. So this enzyme primase, it can make a short RNA primer on this template. These primers are then extended by DNA polymerase. These are acted by the DNA polymerases and they extend the synthesis. So here you can see this is the replication fork and it moves towards this side. These are two single-stranded DNAs and this is primase. And this red colored, you can see, this is a small RNA primer. So this RNA primer is a compulsory requirement for the synthesis of new DNA strand. Both the leading and lagging strands, both these strands, they require primase to initiate DNA synthesis. Both these require the synthesis of RNA primers. Each leading strand requires only single RNA primer. As you know, in the leading strand, once the addition of or the replication or the synthesis of DNA is started, it continues until the replication fork reaches the end of the DNA molecule. So only one RNA primer is required at the leading strand. While on the other strand, that is lagging strand, discontinuous synthesis takes place, which means that new primers are needed for each Okazaki fragment. In this lagging strand, the new fragments are formed, and for each fragment, one primer is needed. And synthesis of lagging strand can require hundreds of Okazaki fragments, and therefore they require hundreds of associated RNA primers. Primase activity is dramatically increased when it is associated with another protein that acts at the replication fork, and this protein is called DNA helicase. DNA helicase unwinds the DNA at the replication fork. Because you know the DNA molecule is helically coiled. So if this fork keeps on moving, it requires that this winding should be unwind. So it creates a single-stranded template that can be acted by the primase. So this the presence of this enzyme DNA helicase, it helps the DNA primase to synthesize new primers.